Lab 6, Passive Transport. Part 2. Membranes are selectively permeable. Diffusion, as mentioned before, occurs when a solute moves from a region of high concentration to a region of low concentration. Movement of a solid down its concentration gradient is another way to put it. A concentration gradient is created when one side of a cell has a higher solute concentration than another. For example, a cell placed in a solution that has a higher solute concentration outside of the cell will cause the solutes to travel into the cell. Diffusion occurs spontaneously and is influenced by the second law of thermodynamics, which states that the transfer of energy from one form to another increases entropy, which is the degree of disorder in a system. Something else the second law states is that the universe is always moving towards entropy. If equilibrium is attained, the cells become useless. When solute concentration is equal on both sides, molecular movement ceases because the solutes are not driven to balance themselves. Therefore, entropy is reached. Energy transformation involves an increase in entropy since the solutes use energy to cross the plasma membrane. Solutes in a cell can go against their concentration gradient using energy to prevent a cell from reaching equilibrium. Okay, so for this experiment, we started out by folding and clipping a 25 centimeter dialysis tube. We placed the open end of the tube over the stem of a clean funnel to fill it with 25 milliliters of starch Na2SO4 solution. We then folded and clipped the open end of the dialysis tube, removing as much air as possible. We then rinsed off the bag in a pan of DH2O solution and patted it dry before submerging it into a beaker of NaCl that acted as the extracellular fluid. We allowed the experiment to run for an hour, and we labeled 8 test tubes. After the 60 minutes were over, we poured 20 milliliters from the bag or dialysis tube into a graduated cylinder and used this to measure out 5 milliliters into the first 4 test tubes. So 5 milliliters per test tube. We then cleaned the cylinder and then poured 20 milliliters of the beaker solution into the graduated cylinder so that we could pour exactly 5 milliliters of this solution into the remaining 4 test tubes. So here we have some pictures and videos of the preparation and completion of the dialysis tube. So we'll start out by watching the video.
We're pouring 25 milliliters of starch in NaCO4. Yeah, SO4. Yeah, SO4 solution into our 25 centimeter tubing.
Once we were done filling the tube, we folded it over and clipped it, as mentioned before, and the middle picture shows how it looked right before we put this cell in its extracellular fluid, or beaker of NaCl solution, which we see on the left. Sorry, I forgot to introduce the video, but that was a starch test for test tubes 1 and 5, and yeah, that was Rachel adding the Lugol's reagent to the test tubes. And here on the right, we have the tubes after we were done adding the reagent, and keep in mind that the test tube on the right will always have contents from the beaker, and the test tube on the left will always have contents from the bag or dialysis tube. So here are the other tests that we performed. We tested for sulfate ions on tube 2 and 6 with 2% BaCl2 solution, and that's the picture comparing the two tubes. And we tested for chloride ions on test tubes 3 and 7 with silver nitrate. Last but not least, we performed a protein test using Biuret reagent on test tubes 4 and 8. Instead of a picture, we actually have a video comparing the two tubes. So we will talk about whether the tests were positive or negative further along in the presentation.
So here are our test tubes in order after adding each respective reagent. And then here is a picture of the test tubes while we're comparing them. And we can start with test tubes 1 and 5. Okay, so here are the results we can compare. Remember that the left side of the bag has the bag content and the right side has the beaker content. So bag and then beaker. Okay, so the Lugol's reagent that tests for starch yields a dark blue-black color in its presence. And test tube 1 has tested positive and test tube 5 tested negative for the starch. And therefore the bag contains the starch while the beaker does not. Now we can talk about test tubes 2 and 6. So 2% BaCl2 is the reagent that we use to determine if sulfate ions were present. And the positive result is recorded if a white precipitate, which actually is barium sulfate, if it forms. So in this case, both of the test tubes were positive. And since you can see slight precipitate formation in test tube 6 and then the obvious precipitate in test tube 2, we can tell that the precipitate had formed. So now we know that both of the bag and the beaker contain sulfate ions. Now it's time to compare test tubes 3 and 7. They were tested with silver nitrate to detect chloride ions, and if a milky white precipitate, which is silver chloride, formed, then there was a positive test. And this shows that both of the test tubes, and therefore both the bag and beaker, yielded positive results for the chloride ions. Lastly, we'll compare test tubes 4 and 8. The BRA reagent that we use tests for proteins, and a positive test will yield a light lavender color. Now we can tell that test tube 4 was negative, while test tube 8 was positive. And this means that the bag did not contain proteins, but the beaker did. So here we have provided a final chart that shows all of the test tubes' results. And the bag's contents are expressed in test tubes 1 through 4, while the beaker's contents are expressed in test tubes 5 through 8. Okay, so really quickly, we'll just talk about sheep's blood. So we were analyzing 0.9% sheep's blood under a microscope. And now we can label each thing as what it was. So here is the isotonic, and you can tell because you can't really see very much. And that's really the point, because in an isotonic reaction, it's not like the water is flowing in or out of the cell, because the solute concentration is pretty much equal on the inside of the cell and the outside environment. So this makes sense. Then you can look at this picture, and you know it's hypotonic because the cells are swelling, and that occurs because the inside of the cell has more solute concentration. This is also just a little picture of an air bubble that we saw. Looked kind of cool, so we thought we'd put it in. Alright, and then here's the last slide we saw, and this is hypertonic. And you can tell because if you look closely, you can see that the cells are shriveling up, which is called crenation, whereas if the cells are bursting, that's called lysis. And it only really shrinks when the concentration of the outside environment solute is greater than that concentration of the inside of the cell. So that's the end of the video, and so we'll see you next time.